Good evening, everyone. So today my topic will be the new frontiers in the management of non-Hodgkin's lymphoma. So I'll be focusing on the B-cell lymphomas since that's where most of the advances are happening and would really push the frontiers for personalized and tailor-made therapy for the patient. So relapse and refractory B-cell lymphomas are a major treatment challenge, as we can see in our patients who come to our clinics. And a significant proportion of these patients seem to be refractory to rituximab at the time of relapse, which is of concern. And many of these patients have poor outcomes at relapse due to inabilities to tolerate toxicities due to the therapy. So novel approaches has been designed to avoid toxicities and target the disease by entirely different approaches. The more effective chemotherapy regimens, new monoclonal antibodies, radioimmunotherapy, adoptive T-cell immunotherapy have greatly improved lymphoma management in the last few years. Since malignancies evade the immune system through alteration of surface antigen expression and T-cell exhaustion, so these mechanisms have really been used to target these malignancies. And the success with immunomodulation has been seen with allogeneic stem cell transplant, when graft versus leukemia effect shows the way for the adoptive T-cell therapy and immune cell targeted monoclonal antibodies. These approaches are really promising in the management of relapsed and refractory lymphomas, some of which may be really heavily pretreated and which otherwise is an area of dismal prognosis. Coming to the first therapy, which really sparks a lot of interest whenever it is spoken, and we are really unlucky that we don't have it in India yet. So CAR T cells, as we know, are autologous T cells which are modified to express chimeric proteins, which are expanded in vivo and then reinfused into the patient. These CAR T cells can recognize tumor antigens independent of the major histocompatibility complex and activate T cells, leading to tumor cell death. And co-stimulatory molecules CD2841BB for T cell proliferation, survival, and persistent anti-tumor effect are integrated into the CAR T cells. So the development of CAR T cells has been going on since the last few years. Well, the first designed around 1993, but it did not have the advantage of persistence in the body and for continuing action. And that's when second generation CAR T cells and the third generation CAR T cells took place. And in 2002, we found the second generation CAR T cells, on which actually majority of the approvals of FDAs have come around 2017, in which Tisgen Leclusil was approved for CD19 CAR, which was approved for pediatric ALL and non-Hodgkin's, B-cell non-Hodgkin's lymphoma. So a brief diagram about how actually CAR T cells are generated. So usually these T cells which are collected from the patient are usually modified by introducing a lentivirus or a retrovirus vector containing the CAR T cell construct, which has a variable chain, which is the outer membrane portion of the T cell. It has the transmembrane domain, which lies here, along with the intracellular T cell activating domain, which is coded by the CD28 and the delta chain. So this is actually then transfused into the patient, and then these T cells into which we introduce this CAR T cell vector starts expressing the chimeric antigen receptor, which recognizes the targeted tumor cells and causes T cell activation, causing in death of the tumor cells. So over a period of time, we have produced various generations of CAR T cells. As I told you, the first generation CAR T cell just had the extracellular domain in which the antigen was recognized, the transmembrane domain which conducted the impulse into the intracellular domain, causing T cell activation. But it did not have the advantage of second generation CAR T cell, in which a post-stimulatory domain of CD28 was integrated, causing persistent T cell activation and interleukin production, which maintains the T cell in the circulation for its action to go on. Because as you know, T cells are terminally differentiated differentiated cells. So in the absence of a co-stimulator or an interleukin like interleukin-2, they will exhaust from the circulation. As and when our knowledge improved in the production of CAR T cells, we started integrating more advantageous molecules like integrating another co-stimulatory signal that causes the same T cell activation but more potent. In the fourth generation CAR T cells, we started integrating new genetic sequences that would cause additional interleukin production that will cause proliferation and maintenance of T cells in circulation. And the fifth generation CAR T cells act also on other pathways of tumor proliferation, like the JAK-START pathway, causing their inhibition. So additional ways in which you can control the tumor proliferation and tumor signaling. So going through the presently licensed products for CAR T cells, we have the TISGen lecalucil, which is licensed for pediatric B-cell ALLs and non-Hodgkin's lymphoma. And it has also been tried quite successfully in DLBCLs and follicular lymphomas with quite impressive overall response rates in relapse refractory settings in which patients are heavily pretreated and expectancy of life is quite low. There are other molecules targeting CD19 like axicabtagene ciloleucel, which is the Yescarta product, which was quite successful in the ZUMA-1 trial with overall response rate of 83% and a complete response rate of 58%. Lisocabtagene maraleucel is another product which is quite effective in diffuse large B-cell, primary mediastinal B-cell, follicular and transformed follicular lymphomas with good overall response rate and complete response rates. So as and when we give CAR T cells, we do recognize that they come with their own problems like cytokine release syndrome, which is present in about 10 to 15% of patients, and neurotoxicity, which is related to the cytokine release, and these really remain a challenge. So as and when our knowledge progressed, we started redesigning the CAR T cells so that the rapid T cell expansion and the systemic perturbation of immune system is corrected. So to mitigate that risk, we started modifying the endogenous CD3 complex and redesigning the T cell activating receptor so that the cellular response of proliferation and the immune system perturbation is actually a bit dampened. So these redesigned CAR T cells have less propensity to cause cytokine release and B cell exhaustion. So the next thing in the market is bispecific CAR T cells. So it was seen that why not target two antigens in place of targeting only one antigen. So there are multiple phase 1 and early phase 2 studies which usually show good efficacy if you target both CD99 and CD22, which are consistently expressed on the B cells. There are CAR T cell studies in which CD19 and CD20 both have been targeted in relapse refractory settings. And there is a phase 1 study currently undergoing in which CD19, CD22 bispecific CAR T has been combined with a consolidation therapy with anti-PD-1 pembrolizumab. So the next thing on the horizon are armored CAR T cells. So it tends to help to mitigate the risk of lack of persistence and expansion of CAR T cells by blocking the immune suppressive environment. So it has been engineered with both the co-stimulatory molecules CD28 and 4-1BB. So it's essentially the second generation CAR T cells. The next thing on the horizon is antibody coupled T cell therapy. So in this CAR T cell construct, what happens is CD16 has been integrated into the CAR T cell, the antigen receptor. So what happens is we give an antibody like rituximab or trastuzumab, which targets the antigen, the tumor antigen, and once it has coupled with the tumor antigen through the CD16, the FAB portion of the antibody is recognized by the CAR T cell, and then it adds to the T cell activity through this coupling and causes intracellular downstream signaling. So this is a summary of whatever I spoke about the new methods of mitigating the toxicity of CAR T cells and also increasing the efficacy, that is bispecific CAR T, the armored CAR T cells, and the antibody coupled T cell receptors. So after the CAR T cells, although it preceded the CAR T cells, the use of bispecific T cell antibodies has been encouraging in non-Hodgkin's lymphoma. They have a documented efficacy in acute lymphoblastic leukemias. So Polatuzumab, as we all know, is a T cell engager. We simultaneously link CD3 and CD19, and it synergizes and complements the T cell mediated cytotoxicity with antibody dependent cellular toxicity. It has been used with impressive single agent activity in follicular, mantle cell, and DLBCL relapsed refractory disease. Mosunetuzumab, which targets the CD3 epsilon and CD20 receptors, has also been shown to have good benefit. The next mode of therapy of interest are antibody drug conjugates. So these combine the cytotoxic potency of a chemotherapeutic agent to a target-specific monoclonal antibody. So there are various molecules which are currently under investigation, like ADCT-402, which targets CD19 and SG3199. It targets CD19 and delivers SG3199, which causes DNA cross-linking, and it has been seen to be strongly potent in CD19 expressing cells. ADCT-301, that is camidanlumab decirin. So it utilizes CD25 to tag these cells and delivers the PBD toxin, and it has been used with encouraging results in heavily pretreated Hodgkin's, PTCLs, CTCLs, and NHLs in which CD25 is consistently expressed. This is a summary table of the bispecific antibodies and the antibody drug conjugates. It has been seen to be used in a diverse population of non-Hodgkin's lymphoma patients with impressive overall response and satisfactory complete response rates. Polatuzumab Vedotin, as we all might have heard, is a CD79B directed antibody drug conjugate, which has been indicated in the combination with bendamustine and rituximab for adult patients with relapsed refractory diffuse large B-cell lymphoma after at least 2 prior therapies. It delivers monomethyl auristatin, a microtubule inhibitor. It has an acceptable side effect profile with peripheral neuropathy and CR rate of 40% and an overall response rate of 63% when combined with bendamustine and rituximab. Unfortunately, it's still not available in India too. This is the JCO article, which showed a definite improvement with polatuzumab and bendamustine in progression-free survival and in the overall survival. There are other experimental therapies which are also currently under investigation, that is macrophage-mediated phagocytosis, in which CD47 is utilized for inducing phagocytosis of the tumor cells through macrophages by unmasking pro-phagocytic signals, and engineered toxin antibodies in which cancer cells are targeted by immunotoxin scaffold with an antibody binding domain. We all know immune checkpoint therapy, and we have very good experience in solid tumors with in immune checkpoint therapy and in Hodgkin's lymphoma. And such similar experience has also been seen when it has been used in relapsed refractory Hodgkin's lymphoma, as in the solid tumors. So since there is high frequency of 9p24 gene alteration and increased PD ligand expression, so it makes classical Hodgkin lymphoma quite responsive to the immune checkpoint therapy. And the rare 9p24 and poor PDL1 expression in B-cell lymphomas are actually is because its use is not too much in non-Hodgkin's, but there are few trials in DLBCL in which these have been combined with other antibodies and targeted agents like lenalidomide and copacillin. Small molecule inhibitors like the PK inhibitors ibrutinib and acalabrutinib have been approved already for CLL, mantle cell lymphoma, marginal zone lymphoma, and Waldenstrom's. And there are other investigatory products like M758, which is a highly potent selective second generation inhibitor, We have phosphoinositol 3 kinase inhibitors, which have been used in similar settings, and some other experimental products, which are also in the pipeline. So this is a summary table of the small molecule inhibitors, which are currently being used and having some response in non-Hodgkin's lymphoma. These are my references, and I'm open to questions. Thank you so much.
So thank you, Roshan, for finishing on time, and I think we have some time for questions. So any questions from the audience or the panelists? Now's the time to ask. But while we wait for questions, Roshan, I think that was a great presentation. I think Dr. Subhash has a question.
Yes, Dr. Subhash.
Subhash, you're on mute. So can you unmute?
Can you hear me now?
Yeah. That's better.
That was an excellent presentation, Roshan. Thanks for that.
Thank you, Dr. Subhash.
Yeah, and I've heard that Polatuzumab is available on compassionate use in India with, who's that, Roche, is it?
Yeah. So as of now, Polatuzumab is not currently available on compassionate, since we found out for 1 or 2 of our patients. So they're not supplying. So maybe around 1 year back, it was available.
Yeah.
Rajiv Gandhi Institute did give it to around 2 or 3 patients, but it's not currently at the present situation. It is not available, and specifically because of the import restrictions, it is not coming in.
Right.
So we were also offered Pola at one point about a year ago, but because of legal problems, I think we couldn't open that compassionate use program in Vedanta. But Polatuzumab was available for the indication that for relapsed lymphomas who were non-transplant eligible, so the older relapsed lymphomas, it was available, and in combination with bendamustine rituximab. So I think what they wanted was Roche wanted to introduce Pola, and they wanted some sort of data to present to DCGI. So they were giving it on a compassionate basis for this specific indication. But they pulled it now, and it's about to be introduced in the open market.
But I mean, the major use would be in, again, relapse post autograft rather than in...
the initial trial was actually for patients who are not eligible for autograft. So I think that is that is how they got the FDA approval, because
that's the indication that they want to market it in India for, in combination with bendamustine rituximab in relapsed lymphomas who are not eligible
for stem cell transplant.
Right. So what's your experience so far in using a combination of ibrutinibs and chemotherapies in refractory DLBLs?
So I feel it is, I have not treated much in that way, but anecdotal experience of use in maybe few patients, but we haven't yet noticed any. I recently put 2 of my patients, but I cannot say that it is really effective to combine ibrutinib with this one, and not much impressive. That's what I think.
Okay.
So fortunately, we're running on time. I can allow one more question. If there are none, then that is good news. Roshan, you've done a great job, which means there are no questions. There was a lot of CAR-T in your presentation, but unfortunately, we have no access to it. So really, a comment.
It sounds very exciting, and once you read into it, it actually spurs a lot of interest that this can be done, so less side effects, and the persistence of CAR T cells and B cells really encourages that there can be long-term responses also in CAR T cells. So obviously that's very exciting, and I hope that it comes.
Unfortunately, it's very expensive and not available at the moment, and that's the main problem. Access to it, I don't know when, but there'll have to be some sort of decentralization into CAR-T. production for it to be effective.
Yeah.